Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 2 from our Brock Biology of Microorganisms 15th edition textbook. This chapter covers microbial cell structure and function, so let's go ahead and get started. Now, one thing we need to discuss with cells is their morphology. Morphology refers to a cell's shape. And when we're talking about prokaryotic cells, like bacteria and archaea, they both have coccus shape, which means spherical or ovoid shape, rod shape or bacillus shape, which is a cylindrical shape, and spirillum, which is a curved or spiral shape. Both bacteria and archaea have this kind of shape. However, bacteria have a lot more diversity in the types of shapes that they possess. They can have appendages. They could have a filamentous form, and they could have vibrio shape. Vibrio would be a comma shape. And then in addition, we could look at the arrangement of the bacterium as well. Arrangement meaning, do those cells appear as chains, like almost like a sausage links, chains of bacterium? That's called strepto arrangement. So chains of spherical bacteria would be known as streptococcus. Or do those bacteria perhaps appear like grapes, a grape-like appearance, where there are clusters of cells? For example, if there are clusters of spherical cells, we would call that staphylococcus. You can see here some of those shapes, the coccus-shaped bacterium. Here's a cartoon on the left, and then an image on the right, phase contrast photograph on the right. You can see what we're talking about, coccus shape, and plural for coccus is cocci. You could see the rod shape or bacillus-shaped bacterium here. Spirillum, spiral shape. Spirochetes, which are corkscrew-shaped. And then these are the appendaged bacterium I told you about, where they could have a stalk or an appendage hypha. And then the filamentous bacterium as well. These actually look like mold when you look at the colonies themselves. So they're really interesting. However, just realize that morphology typically does not predict what the physiology, the ecology, the phylogeny, or other properties of the prokaryotic cell are. You can't predict what a cell does just by its morphology. It can help in identifying a species, but it's only one part of that identification, and the morphology doesn't always give away its physiology. Now, there may be some selective forces involved in setting the morphology. So for example, you could have an optimized cell size for nutrient uptake, optimized surface to volume ratio. I'm going to show you how cell shape affects surface to volume ratio in a minute. It could affect swimming motility. For example, the spirochetes are spiral shaped, and a lot of them have this twisting ability where they can corkscrew through viscous materials, right? So their spiral shape allows them to corkscrew. And then you can have gliding motility. The filamentous bacterium exhibit a type of motility called gliding motility, which we'll explain later on. Now, keep in mind that prokaryotes have a much smaller cell size than eukaryotes. Prokaryotes are about 0.2 micrometers to, some of the bigger ones are really big, but these are the rare exception, to greater than 700 micrometers in diameter. Whereas eukaryotes, look at this, eukaryotes are 2 to greater than 600 micrometers in diameter. Typically, a bacterial cell is about 50 times, 10 to 50 times smaller than a eukaryotic cell. And it says here that most cultured rod-shaped bacteria are between 0.5 and 4 micrometers wide. So, this is a small cell. And this is why in the laboratory with the light microscopes, when you look under the light microscope, those cells are so small. Even at 1000x magnification with oil, those cells are very small. And here on this table, you can see clearly the different bacterium and the different sizes, their associated sizes as well. Now, I mentioned earlier about surface to volume ratios, how the cell size affects surface to volume ratio. And this is important because if you have a greater surface to volume ratio, you have greater nutrient and waste product exchange per unit cell volume. So waste can get out of the cells easily, nutrients can get into the cells easily, they tend to grow faster, they could lead to faster evolution. And you can see here that you have a smaller cell. This small cartoon represents a smaller cell versus a larger cell. And if you were to look at the surface area by volume ratio, smaller cells have a larger surface area by volume ratio. They have a surface area to volume ratio of 3, for instance. Whereas larger cells have a smaller surface area to volume ratio. And again, why is this important? Well, this allows bacterium to grow as fast as they do, to take up nutrients as fast as they do, to secrete wastes as fast as they do. And that's really what allows them this competitive edge of being so small. All right, moving on to the cell membrane and cell wall. We have to start our discussion with the cytoplasmic membrane. Remember that all cells have a cytoplasmic membrane, but the composition of that membrane and some of the functions of that membrane may be different. The membrane surrounds the cell. It surrounds the cytoplasm. It's what keeps the cytoplasm inside of the cell and separates the cytoplasm from the environment. The main reason we have a cell membrane is its selective permeability, which means that it allows nutrients to be transported in, waste to be transported out. And the general structure of the bacterial membrane, the bacterial cytoplasmic membrane, is a phospholipid bilayer with embedded proteins. You can see that here. This is the phospholipid bilayer. Much like the eukaryotic membranes, you have phospholipids. Phospholipids have a glycerol head, a phosphate group, and then some kind of attachment to the phosphate group. This could be choline, this could be ethanolamine. There's different attachments here, positively charged. And then you have your 2 fatty acid tails. And these phospholipid units form a bilayer with the heads pointing outside of the cell because the heads are charged and hydrophilic, with the tails pointing inside the middle, into the middle of the cell membrane, because those tails are hydrophobic. So because of that, you have what's known as the hydrophobic region of the plasma membrane, and that is where the tails live. And then you have the hydrophilic portion of the plasma membrane. That's where the heads are. And again, the heads point to the fluid outside of the cell and to the fluid inside of the cell. And here is a electron micrograph of the bilayer right there. You can see the bilayer with the fatty acids in the middle. So to further explain the structure of the plasma membrane, realize that it's very thin. It's only about 8 to 10 nanometers wide. It can be strengthened by these sterile-like molecules called hopanoids, kind of like our human Plasma membranes have cholesterol, and cholesterol helps to strengthen the cell membrane and keeps fluidity and serve as a fluidity buffer in the cell membrane. Bacterial cells have hopanoids, and these are sterols in the membrane that keep the membrane strengthened. Okay. In addition to that, obviously you have many other proteins embedded in the plasma membrane. Some are integral membrane proteins, which means they're significantly embedded, and some are peripheral membrane proteins, which means they're loosely embedded. You can see here, this is the plasma membrane, the lipid bilayer, and you can see all these different proteins are embedded in the plasma membrane. Some are peripherally embedded, loosely embedded, and some are integral. They are significantly embedded in the plasma membrane. And what are the functions of these membrane proteins? There's a myriad function for these membrane proteins. They could serve as transporters. In fact, this one is probably a transporter because it has a hole in the middle. They could serve as transporters. They could serve as receptors. They could serve as identity markers. They could serve in cell-to-cell contact. They could serve as part of a metabolic pathway. There's many functions for these membrane proteins, and there are many different membrane proteins. What about archaeal membranes? Archaeal membranes have an ether linkage in the phospholipids, whereas remember that in bacteria and archaea, there's an ester linkage in the phospholipids. Archaeal membranes also look a lot different. They have isoprenes instead of fatty acids. I'm going to show you what that looks like in a little bit. Major lipids are phosphoglycerol diethers with phenyl C20 side chains and diphosphoglycerol tetraethers with biphenyl C40 side chains, which can form lipid monolayers. So check this out. This should look a lot different than the composition of the eukaryotic or bacterial membrane. Here you have your glycerol diether. That's an ether linkage. Remember, there's an ester linkage in eukaryotes and in bacteria. And here is your diglycerol tetraether. You see, it's a tetraether because you've got 4 of these ether linkages, and these will form a monolayer. Because notice how the hydrophilic heads are on both sides, and the tails they span between the 2 hydrophilic heads. So here, if you look closely here, this is called a monolayer because this hydrophilic head and tails, these 2 tails and this hydrophilic head are all connected. Everything's connected. So there's no bilayer here. So you can have a lipid bilayer, or you could have a lipid monolayer. You could have C20 or C40. Okay, in A you have the C20, so a shorter tail, and in B and C you have C40, which is the longer tail. Very interesting stuff. So what you should take away is that archaeal membranes use ether linkages instead of ester linkages. They have, the lipids are isoprenes instead of fatty acids, and you can form a monolayer because of these tetraethers instead of a bilayer. Those are the takeaway messages from the archaeal membrane. And what are the functions of the cytoplasmic membrane in these prokaryotic cells? Well, just like in eukaryotes, it's a permeability barrier, which means only nutrients in, only waste out. It can select what comes into the cell. It can select what goes out of the cell. Remember that polar and charged molecules must be transported. Because the plasma membrane is nonpolar, that means that polar and charged molecules cannot simply cross the bilayer. they need to be transported through a transporter. So only small nonpolar molecules can cross the lipid bilayer. Polar or charged molecules need a transporter in order to cross the membrane. Transport proteins accumulate solutes against the concentration gradient. So you can have active transport as well through these transporters of substances against the concentration gradient. Let's say that a cell wants a lot of sugar inside. It could take up sugar actively using energy, and it can have a higher concentration of sugar inside than outside. Okay, the membrane serves as a protein anchor. Remember the integral proteins and the peripheral proteins, all those proteins anchor to the membrane. And the membrane can serve as for energy conservation and consumption. You can use the membrane of prokaryotic cells to produce ATP. That's where ATP synthase and the electron transport chain live. You can use the proton gradient that's formed by the electron transport chain to do work across the membrane. So in prokaryotes, the membrane is utilized for energy purposes. So you can see that highlighted here. The membrane is a permeability barrier. It is a protein anchor for all these membrane proteins, and it's used in energy conservation. It drives a proton gradient, so you get a high concentration of protons outside due to active transport of protons with the electron transport chain, and then the bacterial cells can utilize that proton gradient through facilitated diffusion in order to do work, in order to make ATP, in order to run the flagella, in order to transport molecules across the membrane through transporters. So bacterial cells are neat because they can use this proton gradient that they make in order to do lots of work, in order to do cool things like propel themselves or make ATP, right? So they're really versatile in that way. Now recall that bacterial cells have cell walls, typically have cell walls, and they can be separated into two groups based on their cell wall architecture. There are bacteria that are considered gram-negative cell wall bacterium, and these are ones that stain negative or stain pink in the gram stain procedure. And then there are gram-positive cell wall architecture bacterial cells that stain positive, stain purple in the gram stain procedure. And it all has to do with the architecture of the cell wall. Gram-negative bacterium have two membrane layers, two cell membranes. And then they also have LPS, lipopolysaccharide, and peptidoglycan in their cell membrane as well. So I'm gonna go through in detail and explain to you what this means, what the membrane of gram-negative cells looks like. Gram-positive cell walls, on the other hand, look a little bit different. There's only one cell membrane, but the cell wall itself is made of peptidoglycan, and it's thick. It's much thicker layer of peptidoglycan, and there's not much more to it than that. You have a thick layer of peptidoglycan as the cell wall. You can see it here. On the left, you have a gram-positive cell with a gram-positive cell wall architecture, and on the right, you have a gram-negative cell with gram-negative cell wall architecture. Again, gram-positive cell stain purple in the gram stain, and gram-negative stain pink in the gram stain. So take a look here. With the gram-positive, if we were to zoom in on the cell wall here, you have a plasma membrane. This is your plasma membrane with a phospholipid bilayer. Then outside, you see this structure here. This is a thick layer of peptidoglycan. Peptidoglycan is the cell wall material of bacteria, and it is unique to bacteria, which means archaea do not possess peptidoglycan and eukarya do not possess peptidoglycan. It is strictly a bacterial thing, and most bacteria possess a cell wall of peptidoglycan. Then look at here. You have your gram-negative cell wall architecture, and remember I said there are two cell membranes. Here's your cytoplasmic cell membrane here. So the cytoplasm would be down here. And then you have a thin layer of peptidoglycan. You see this? This is a thin layer of peptidoglycan compared to gram-positive's thick layer of peptidoglycan. And then outside of that, you have a whole nother membrane, a whole nother cell membrane bilayer. And that's called the outer membrane, the outer membrane. So what is the structure of peptidoglycan? What does this membrane cell wall material look like? What is it made of? Well, first of all, it provides a rigid layer to strengthen the cell wall. It's the reason cells retain a shape. You know how there are coccus-shaped bacterium and bacillus-shaped bacterium. There's spirilla. Well, it's the cell wall that keeps the bacteria in those shapes. And it's typically composed of alternating modified glucose. So it's a modified form of glucose called N-acetylglucoseamine and N-acetylmuramic acid. So you have two sugars, two different sugars, and they're linked together into a disaccharide. They're linked together by beta 1,4 linkages. That's the way, that's the bonding, that's the name of the bond that forms between these two sugars. And then attached to the sugars are amino acids, L-alanine, D-alanine, D-glutamic acid, and either L-lysine or diaminopimelic acid, known as DAP. Okay, so again, you have two sugars attached by beta 1,4 linkages with a small peptide comprised of these amino acids attached to that. And I'm going to show you that in the next figure. And then those cross-link differently in gram-negative bacteria and gram-positive bacteria. So the way these peptides cross-link with one another in adjacent strands of peptidoglycan varies between gram-positive bacteria and gram-negative bacteria. So here is a unit. This is a building block. You could call it a building block for peptidoglycan cell walls. This is a peptidoglycan cell wall unit, subunit. Again, remember you have two sugars. These are your two modified glucose. This one's called N-acetylglucoseamine. This one is called N-acetylmuramic acid. And look how they're linked together. Remember, they form a disaccharide through a beta 1,4 linkage. Okay, that's the glycosidic linkage there. And then attached to N-acetylmuramic acid, you have the peptide chain. Remember the amino acids? You've got L-alanine, D-glutamic acid, diaminopimelic acid, and D-alanine. There you go. So you have your short peptide tail. It's called the glycan tetrapeptide. Glycan tetrapeptide. All right. Now, if you take a look here, remember I said adjacent strands of peptidoglycan. So here's a strand of peptidoglycan, and here's a strand of peptidoglycan from E. coli. Notice how DAP cross-links to D-alanine on the adjacent peptide strand, and there is no bridge that forms between. It's just a direct linkage. So you can see here no interbridge is present in E. coli peptidoglycan, nor in that of other gram-negative bacteria. So in gram-negative bacteria, DAP will, for example, directly bind to D-alanine. And so this strand of peptidoglycan can directly bind to this strand of peptidoglycan, forming these crosslinks. You see these little crosslinks. On the other hand, with gram-positive bacteria, such as Staphylococcus aureus, you form a glycine crosslink, right? so look here, the glycine interbridge or the glycine crosslink in the gram-positive bacterium S. aureus. So in this case, L-lysine is bound to glycine bridge or interbridge to dealanine on the adjacent strand of peptidoglycan. And this is really interesting. In figure C, you can see the overall structure of peptidoglycan. So you have these adjacent strands of peptidoglycan that kind of look like this. You see, peptidoglycan strands form a spiral like this. And these are the little peptide chains that hang off. You see, these are the little peptide chains. So when you have the sugars form a spiral. The little peptide chains poke out the sides, and when they're adjacent to one another, these peptides helices are adjacent to one another, they can form these cross bridges with one another. So note how the glycosidic bonds confer strength on the peptidoglycan in the X direction. So the glycosidic bonds, that's this guy, the beta-1,4 bonds, the beta-1,4 bonds confer strength on peptidoglycan in the X direction and in the X direction, whereas the peptide bonds, the interbridge or the peptide bond between DAP and dealanine, for instance, those confer strength in the Y direction. Did you see that? So the cell wall has strength in this direction due to those sugar bonds, the beta-1,4 bonds. And there's strength to the cell wall in this direction due to these interbridges I told you about. Remember, gram negatives don't have an interbridge. They link the adjacent peptide strands directly, and gram positives do have a glycine interbridge, because they don't bind directly. And this is, again, what one strand of peptidoglycan looks like. It looks like a helical structure with a polypeptide chain, a short polypeptide chain hanging off. So you can imagine if there's another adjacent helical strand right here of peptidoglycan, these tails could then bond to one another with peptide bonds. They could bond to one another either with a direct bond in gram negatives or with an interbridge, a glycine interbridge with the gram positives. All right, now let's discuss the gram-positive cell wall in a little more detail. Remember, the gram-positive cell wall has a thick layer of peptidoglycan, and it could be up to 90% of the cell wall being made of peptidoglycan. So also very common in cell walls of gram-positive bacteria are teichoic acids, which are acidic substances. These acidic substances, and recall that acidic substances have a negative charge. These negatively charged teichoic acids, they can bind to positively charged metal ions such as calcium, magnesium, prior to their transport. And this can increase the flexibility of the cell wall. Lipoteichoic acids are a little bit longer than teichoic acids, and they actually bind to the plasma membrane beneath. So let me show you an image here. You have the plasma membrane of the cell, and the lipoteichoic acids, you see how long they are? They kind of span from the plasma membrane into the thick peptidoglycan layer and out. And then the regular teichoic acids, they are simply embedded in the peptidoglycan. And notice how thick this peptidoglycan layer is. Again, here you can see how the layout of this thick peptidoglycan is. You have the peptidoglycan cables, and these cables essentially look like this. Remember, they're helical in structure with those NAG-NAM subunits in repeating order. So those cables wrap around the bacterium, and they are very thick. Now, some prokaryotes lack a cell wall. These are known as the mycoplasmas or thermoplasmas. Mycoplasmas are a type of bacteria that lacks a cell wall, and thermoplasmas are a type of archaea that lack cell wall. And because they lack a cell wall, they tend to be a little smaller in size, and they tend to not have a particular shape. So they'll be pleomorphic in shape. So now we're talking about the gram-negative cell wall. Remember the gram-negative cell wall. Only a small amount of the total cell wall contains peptidoglycan, unlike gram-positive cell walls where 90% of the cell wall was peptidoglycan. For gram-negatives, only a small amount contains peptidoglycans. Most of the cell wall is composed of the outer membrane. Remember the second membrane of gram-negative cells. And this outer membrane is comprised of lipopolysaccharide, or LPS. I'm going to show you a picture of that in a second. This is a barrier against antibiotics and other harmful agents. The LPS consists of 3 parts: a core polysaccharide, an O polysaccharide, and a lipid A, also known as endotoxin. It's a very toxic compound. So the LPS replaces most of the phospholipids in the outer half of the outer membrane. I'm going to show you an image of this. And don't forget, the lipid A portion of the LPS is known as endotoxin. It's a very toxic component of the LPS. It's toxic to us humans. This is what LPS looks like, lipopolysaccharide. Remember, there's 3 parts to it. The O side chain, or the O polysaccharide. You've got the core polysaccharide, and then this is the lipid part, lipid A, and that's the part that embeds into the membrane, into the outer membrane. So essentially this sugar part sticks out of the outer membrane, and it's the fatty acids of the lipid portion of lipid A that embed the LPS structure into the outer membrane. So here you can see the gram-negative cell wall. You have the plasma membrane, then you have some space called the periplasm. This is the space between the plasma membrane and the outer membrane. You see the space is called the periplasm. Inside of the periplasm you have a thick layer of peptidoglycan, and then you have your outer membrane. You see this is your outer membrane. And you see these structures that look like squares with hexagonal structures stuck to them? That's your LPS. You see how the LPS only exists on the outer part of the outer membrane, not the inner part of the outer membrane, but the outer bilayer, the outer layer of the phospholipid bilayer is comprised mostly of LPS. So the LPS kind of sticks out into the surrounding media. Okay. There's also what are known as porins here. These are large porous structures that allow fluid to cross the outer membrane. These are called porins as well. Okay, I was just mentioning this. The porins are transmembrane protein channels for entrance and exit of solutes. Solutes can cross the porins in the outer membrane. And remember what I told you about the periplasm, this space here between the inner and outer membranes? Space located between cytoplasmic and outer membranes. It can house many extracellular proteins. So that's all for the cell wall. Let's take a quick little break time with Gizmo and Wicket, and we'll be right back to finish this chapter. What do you say? Alright, welcome back. Let's continue on with our tour of the prokaryotic cells. So here we're talking about capsules and slime layers. Together they're known as glycocalyx. The glycocalyx. These are not considered part of the cell wall because these do not confer significant structural strength, although they are on the surface of the cell. They're actually outside of the outer membrane or the plasma or the cell wall. They're the outermost layer of the cell, but they're not considered part of the cell wall simply because they're not there for giving strength or stability to the cell, they're mainly there for other reasons, which we'll discuss in a second. So these are polysaccharide layers. They could be thick, thin, rigid, or flexible. The capsule is, so again, these are sugary layers on the surface of the cell. The capsule is tightly attached. It's a tight matrix, and it's visible if treated with special negative staining or special India ink. You could stain it, you could make it visible. But usually the way you see capsules is by a negative staining technique where you stain the background in order to see the capsule. So again, these capsules are tightly associated with the cell, whereas slime layers are very loosely associated with the cells and they're easily deformed. So you can see here, there's some figures A, B, C, and D. In A, you're looking at a bacterium that's known for having this slime layer. This is Leuconostoc mesenteroides. Leuconostoc mesenteroides. This is a colony on a plate. And what you're seeing here is a loop. You know how you guys are working with the loops in the lab. This is a sterile loop that they've come and touched the colony. And look how gooey the colony is. The colony is very gooey. It has this very thick, gooey consistency. And that's because of the slime layer formed by the cells. Here in B, you can see Acinetobacter species observed by phase contrast microscopy after negative staining with ink. Remember, so you can see here the capsules of these bacterium. You see that halo? That's the capsule of the bacterium. And with negative staining, what you've done is you've stained the background of the slide. You've even stained the bacterium. So you see this dark color is the bacterium, and you've also stained the background. And what's not stained, these little halos, that's showing you the capsule layer, the thick capsule layer of the cell. Here you see a transmission electron micrograph, so TEM, of a thin section of this bacterium, Rhodobacter capsulatus. It's actually named after its ability to have a capsule, which is neat. And you can see the capsules here with the little arrows. I know the little arrows are hard to see, but the arrows are pointing at the capsule of this bacterium. And here you can see another capsule for another bacterium as well. So these capsules can be thick, they can be thinner, but again, capsules are closely associated and tightly associated with the cell, whereas slime layers are loosely associated with the cells. And what do they do? What do capsules and slime layers do? They can assist in attachment to surfaces. So for instance, you might be wondering, even though you brush your teeth and your teeth are so smooth, how do bacteria keep attaching to that smooth surface and growing? Part of that is these slime layers or capsules allow them to stick, and part of it's also the fimbriae, which we'll talk about in a little bit, but it can assist in attachment to surfaces. It could play a role in the development and maintenance of a biofilm. Biofilms are like little communities of bacterium, where the biofilm is a sugary proteinaceous substance that they produce in order to make a community. In fact, plaque on your teeth is an example of a biofilm. It could be a virulence factor, so it could protect against phagocytosis. So for instance, bacterium that get into your bloodstream, for instance, usually your immune system is able to sniff them out, and phagocytosis, your white blood cells are able to gobble up these bacterium normally, right? But with a capsule, you could actually cloak yourself and kind of evade the host immune system. And it can prevent against dehydration and desiccation. These are little hardy little bacterium because they are resistant to desiccation, which means dehydration. Other structures to be aware of are the fimbriae and the pili. These are filamentous protein structures. The fimbriae enable organisms to stick to surfaces or form pellicles, thin sheets of cells on a liquid surface. So again, I gave you the example of how come bacteria stick to your smooth teeth so easily? Well, part of it is the glycocalyx, the slime layer. Part of it's also these fimbriae, which are these little bristly structures, these filamentous protein structures that serve like little bristles and just like allow the bacterium to stick to smooth surfaces. Pili, on the other hand, are typically longer and fewer. Normally you only have one or a few, whereas with fimbriae you see many. And where do we find the pili? Well, during conjugation, remember that bacterium can conjugate with one another. I may have mentioned that in our lecture, that bacterium can form what's known as the sex pilus, which is a hollow protein tube which extends from the bacterium and can dock on an adjacent bacterium of either the same or different species. And those bacterium can exchange, or it's a one-way exchange, I should say, of genetic information. So the sex pilus during conjugation, also another form of pili is for twitching motility and for adherence to the host tissues. This is the type 4 pili. So here you can see fimbriae are the short bristles on this bacterium. Flagella are much longer. These whip-like structures are flagella, and the short bristle-like structures are the fimbriae. Here you can see two bacterium connected by the sex pilus. The sex pilus being this hollow protein tube that connects bacterium during conjugation. We'll learn all about that in a subsequent chapter. So what are cell inclusions? Inclusions function as energy reserves, carbon reservoirs, and or have special functions. You can think of them as storage sites inside of the cell. They're enclosed by a very thin membrane. They can reduce osmotic stress for cells. They can serve as carbon storage, like glycogen storage in case the cell needs to store some sugar for later, for energy. Okay, and what are endospores? We've mentioned in class several times that some bacterium are able to form endospore. This process is known as sporulation normally, or endosporulation. This is when the bacterium of certain species, mainly, you see here, mainly the genuses that can form endospore are the genus Bacillus and the genus Clostridium. These cells can actually form these endospores, which are highly differentiated cells resistant to heat, harsh chemicals, and radiation. These endospores are resistant to heat, chemical, and radiation. And you could add desiccation, drying out as well. They're very resistant, and this is one of the primary reasons why we have to autoclave our utensils, for our instruments for surgery. We have to autoclave needles for tattoos. There's autoclaved instruments at the dentist's office. We need to use an autoclave because you need that extra heat, that extra pressure in order to destroy endospores. All right, so survival structures to endure unfavorable growth conditions. Yes, so during unfavorable growth conditions, these bacterium can form endospores. And then the bacterium can actually germinate. When the conditions get better, the endospores can germinate, and the vegetative cell, which means the regular cell, can grow out from the endospore. And again, it's present only in some gram-positive bacteria. These endospore formers can form endospores in a variety of locations. Some bacterium are known for terminal endospores, which means the endospore forms at the terminus, at the end of the cell. Subterminal endospores near the end of the cell, or central endospores. These are endospores at the center of the cell. And don't forget this term, vegetative cell. The vegetative cell is the normal growing cell. And the vegetative cell can be converted to a non-growing, heat-resistant, light-refractive structure. Basically, the vegetative cell, when faced with an insult being, chemicals, bad chemicals, so UV, desiccation, lack of nutrients, that can spur on sporulation. So the vegetative cell can start developing into endospore inside. Do you see the vegetative cell feels some kind of insult? The vegetative cell then develops an endospore within. Remember, and it could be different location inside of the cell depending on the type of cell. And then the mature endospore can then be released, and this is your hardy little structure that has the genome inside and can resist so many different harsh conditions. And then when the conditions improve, that endospore, like a seed almost, can germinate to form the new vegetative cell, which can go on and grow and do its job. Okay. So again, this endospore can remain dormant for years, but converts rapidly back to a vegetative cell once the conditions improve. And the way that this happens, the way that a dormant cell can reactivate is through a process: activation, germination, and outgrowth. I'll show you a picture of that soon. But during activation, heat for several minutes at elevated but sublethal temperature, so you need some heat. Germination, it's rapid. Loss of refractility and loss of resistance to heat and chemicals, and then the outgrowth of the vegetative cell, swelling up, water uptake. You need to rehydrate, and synthesis of RNA, proteins, and DNA, and you're going to resume your role as a cell, a vegetative cell. So you can see here in A you have activation, outgrowth, this is the refractive stage, continued outgrowth, and then the new vegetative cell emerges and grows and rehydrates. Okay. What are the structures of the endospore? There's many layers to an endospore. There's the outermost layer called the exosporium, then the spore coat, the cortex, the core. Inside of the endospore you have dipicolonic acid, which actually binds to calcium and dipicolonic acid kind of bind to one another. I'll show you an image of that in a minute. And the core contains small acid-soluble spore proteins, SASPs, which bind and protect the DNA and function as a carbon and energy source for outgrowth. So you can see here, here's an endospore. Here's an endospore with the exosporium outer coat, exosporium, and the spore coat inside. These are protective structures. Then the core wall, then the cortex, usually a peptidoglycan cortex, and then you have the DNA inside. And this structure is highly dehydrated too. It's very dehydrated core here. This core is dehydrated. And that again protects the endospore from dehydration because it's already dehydrated. So it's not susceptible to further dehydration. Here you can see what I was talking about earlier. You can see calcium complexes with dipicolinic acid. Here's your dipicolinic acid. Calcium complexes with the dipicolinic acid, helping to dehydrate the developing endospore. Also, this complex inserts itself into the bases of DNA, helping to stabilize the DNA against heat denaturation. So not only does this help dehydrate the core, but it also protects the DNA from heat. And here you can see the whole process I told you about. You have the vegetative cell. During stage 1, you have asymmetric cell division, and you commit to sporulation. Okay, so the cell has sensed something wrong. Otherwise, if it doesn't, it just grows. The cell divides, right? The cell grows and divides. This is called the vegetative cycle. Cell grows, divides, grows, divides, grows, divides. But if the cell senses a problem, like I said, an insult, well then the cell could decide to sporulate, right? The cell decides to form a spore. So the first step is to form a spore, copy the DNA, and then instead of binary fission, one of the new cells becomes a forespore. There's septum formation. Then there's engulfment, where the plasma membrane there's a second membrane around this spore. Then you have cortex formation, where peptidoglycan coats around the developing spore. Then you have spore coat formation, which is a protein coat. Those SASPs and dipicolinic acid conjugate with calcium to dehydrate the core. The core dehydrates. And then there's maturation, and you have the mature endospore, and then there's lysis of the cell wall, which means that the spore releases to become a free endospore. So there you go. Now you've released the endospore, and the endospore can last and persist through years until the conditions improve, and then the endospore will germinate. Remember, germinate. So this is just a more detailed version of that explanation that you could read through on your own. But again, it's the process of sporulation, step-by-step process of sporulation. Again, the sporulation cycle that I just went over with you is a complex series of events. 200 plus spore-specific genes are involved in this process. Again, not many bacterium can form endospores. Two main genera form endospores. You have bacillus and clostridium, but you could also have, there are a handful of other genera that can form endospores, but the main ones are bacillus and clostridium. Okay, formation of vegetative cell, we already touched on this. Activation, germination, and outgrowth. Remember, this is how the vegetative cell forms from an endospore. Okay, let's talk a little bit now about cell locomotion. So how does the cell get around? How does the cell move about? Recall that not all bacterium or archaea have a form of locomotion. There are several bacterium and archaea that have no ability to move about, okay? But some do, and there's different techniques that they can use in order to do so. So let's talk about a few of them. Flagella, we can talk about swimming motility. We could, well, actually flagella, gliding motility. I don't believe we touch on axial filament or corkscrew motility, but we might in a later chapter. So recall that, flagella is a major way by which some bacterium get around. You've got this whip-like tail, that rotates in order to propel the bacterium. Now, in eukaryotes, the flagella is helical in shape. Okay. Oh, I'm sorry. In prokaryotes, the flagella is helical in shape, whereas in eukaryotes, remember, there's a fundamental difference between eukaryotic and prokaryotic flagella. You need to know this. In eukaryotes, remember that the flagella beats back and forth like a fish tail. You need to know that in eukaryotes, the tail beats back and forth. Think of a sperm cell, for instance. If you've ever seen those microscopy videos of sperm cells swimming, their tail beats back and forth. The prokaryotic flagella is fundamentally different. It's helical and it spins like think of a boat propeller, right? You know what I mean? A propeller on a boat. Imagine attaching a whip to a boat's propeller, right? It literally spins like this, right? So it's spinning, spinning, spinning. Prokaryotic flagella, spin. Eukaryotic flagella, beat back and forth. So they're not the same. They are not the same. And also, again, some some bacterium have no flagella. These are called atrichous. So it would be atrichous bacterium have no flagella. If a bacterium has one flagella, it would be called a monotrichous bacterium. If a bacterium has that one flagella at one end, it's called a polar flagellum, which means the flagellum is at one end of the cell. If the bacterium has a flagella at each end of the cell, that's called a amphitrichous bacterium. If the bacterium has a tuft or a cluster of flagella at one or both ends, it's called a lipoteichoic bacterium. And if the bacterium is completely surrounded by flagella, this is called the peritrichous bacterium. That means there's complete, like look at this. This would be a Proteus vulgaris, which is a bacterium that's completely surrounded by flagellum, for instance. And all of these are possible. Okay. So what does the flagellar structure of bacteria look like? It has many components. The main component that makes up the flagella, the whip portion of the flagella, is called the protein flagellin. And again, the motility is done by rotation, similar to the motion of a boat propeller. The flagella of archaea is about half the diameter of the bacterial flagella, composed of several different proteins, and also moves by rotation. So the flagella of archaea and bacteria are very similar. So here you can see the mechanism by which the flagella functions. Here is gram-negative bacterium. I know because there is the plasma membrane here, and then you have the periplasm, and this is the periplasm. Inside of the periplasm you have the thin peptidoglycan layer, and then you have the outer membrane of the gram-negative bacteria. And notice how this portion of the flagella is embedded in the cell wall. Well, this is called the basal body of the flagella. The basal body includes a stator. The stator, which consists of the MOT proteins that surround the basal body, you see? So you've got the MOT proteins of the stator. This is the stator. And surround the basal body and function to generate torque. The MOT proteins function to generate torque. Torque that literally spins the flagella like a propeller. Now the part that attaches the filament, you know, the long whip-like structure of the flagella, there's a portion called the hook. The hook connects the flagella to the basal body. Okay, so you've got basal body, you've got the hook, and then you've got the long flagella made out of the protein flagellin in bacterium. And the energy, you might be wondering, where does the energy come from? How does this thing spin? The energy comes from the proton motive force. Remember, there's a lot of protons outside of the cell here, and the reason there's such a high concentration of protons outside of the cell is because of remember the electron transport chain that's in the outer membrane, or sorry, it's in the inner membrane of the cell. It's pumping protons out. It's pumping protons out here, and those protons forcing their way back into the cell is what generates that torque. It's what generates the power. It's what generates the power to spin the flagella. Isn't that neat? So what's gliding motility? Well, this is found in bacteria, not in archaea. It's slower and smoother than swimming. The movement typically occurs away from the colony, requires surface contact. So you need to be, you're gliding on something. You have to be gliding on a surface. And what's the mechanism? You excrete polysaccharide slime, and then your type 4, remember your type 4 pili, conducts twitching motility. And then you've got, let me show you here. So here is your surface that you're gliding on. And this is your surface. And these are the glide proteins here. You've got glide proteins. And with the motion of these glide proteins, the cell is moving to the left. The cell is moving to the left while the glide proteins are kind of moving in the opposite direction. See, it says here the tracks in yellow. The tracks in yellow exist in the peptidoglycan that connect the cytoplasmic proteins to outer membrane glide proteins. These are your glide proteins, outer membrane glide proteins. And propel the glide proteins along a solid surface. Notice that the glide proteins and the cell proper move in opposite directions. See, the glide proteins are moving in this direction towards the right, and that allows the cell to move towards the left. Here's some examples of different creatures that can conduct or employ glide motility. You have oscillatoria, which is a type of photosynthetic bacterium. You have Flavobacterium johnsoniae, which is a well-known glider, also gliding. And this one's really neat. This is a mutant strain of Johnsoniae which cannot glide. And notice how, you know, the colonies cannot move about. The bacterium can't move away from the colony. They can't glide away from the colony due to their mutation. But here, the colonies look completely different, and that's because the bacterium are gliding away from the main colony, probably in search of food. So why move? Why move at all? Well, because bacterium are usually trying to get somewhere. They're trying to conduct what's known as taxis. Taxis is directed movement in response to a chemical or physical gradient. Chemotaxis is movement in response to chemicals. So the bacterium, it might be moving towards food or maybe away from a harmful chemical. Phototaxis, response to light. Photosynthetic bacterium, for example, if you're a cyanobacteria, you might want to go towards the light. Aerotaxis, you might want to go either towards or away oxygen. Some bacterium will die in the presence of oxygen, so they got to get away from the oxygen. Other bacteria might want to go towards the oxygen. Osmotaxis, response to ionic strength. Some bacterium want to go towards salty conditions. Some bacteria might want to get away from salty conditions. They'll get dehydrated. And then hydrotaxis, response to water. How do the bacteria go towards the moisture or away from? So again, chemotaxis is directed cell motion towards a chemical, best studied in E. coli, which is peritrichous. Remember what peritrichous means? Completely surrounded by flagella. So this is what peritrichous means. And when you're looking at flagellar chemotaxis, what you're going to see is run and tumble behavior. This is what you're going to see, run and tumble behavior. That's how flagella work. During a run, the bacterium is moving forward, right? There's smooth forward motion. Flagellar motor rotates counterclockwise. If you need to remember this, counterclockwise rotation of the flagella propels the cell forward, okay? And then during a tumble event, remember there's runs and tumbles, during a tumble event, the bacterium stops and jiggles. It kind of tumbles around. The flagellar motor rotates clockwise, so it goes from counterclockwise, propelling the bacterium forward. CCW means counterclockwise. Counterclockwise propels the bacterium forward during a forward run. Clockwise rotation, so it's spinning now the other way, causes tumbles and stops and jiggles, right? The flagellar bundle can come apart. The flagellar bundle come apart. If it's peritrichous, you have the tumble, and look at all the flagella coming apart. During a run, the flagella kind of, they all kind of like bundle together. And again, they move by rotation. So you can see here through run and tumble, every time it tumbles, it changes direction. During a run, the bacterium is moving straight, but during a tumble, it's changing direction. You see this? So bacterium are running and tumble. So run, tumble, run, tumble, run, tumble, run, tumble. If there's no attractant, if there's no chemotaxis going on, the motion is random, which means the runs and tumbles aren't really getting you anywhere. However, if there is an attractant, like let's say there's sugar out here or something tasty, the bacterium will still run and tumble, but eventually it will make its way towards the attractant. Does that make sense? So you're still running and tumbling, and at times it does look like you're getting away from the attractant, but you soon change course, and your net migration is a directed movement towards the attractant. Okay. So what's chemotaxis like in polarly flagellated bacterium? Remember in peritrichous bacterium, during a run, the flagella kind of bunch up. During a tumble, they split apart. So polarly flagellated bacterium, where you have one flagella, is similar but not identical to peritrichous motility. Okay. Many, including Pseudomonas, can fully reverse flagellar rotation, avoiding tumbling and reversing direction. So if you spin the other way, it just takes you the other way instead of tumbling. That's kind of interesting. But some, like Rhodobacter, stop and are affected by Brownian motion. Brownian motion is not a true form of motility. It's a false motility, also known as a fake motility. But it looks like motility because the bacterium is kind of bouncing around. But the reason for the bouncing around is because molecules of mainly water and solution are kind of moving the bacterium around. Think about, imagine if you were in a boat in a like a lake or something, and the lake's really upset, right? Your boat will kind of move around in the lake, you know, but it's not because your propeller's on, you know, your motor's on, or your sails are up. It's just the boat's kind of moving about due to the water. So Brownian motion is not a true form of motility. Okay, so again, in peritrichous motility, during counterclockwise rotation, the flagella bundle. During a tumble event, the flagella are pushed apart, and there's clockwise rotation of the flagella. And during a tumble, you're changing directions until your next counterclockwise rotation, and you move in a different direction. With polar flagella, counterclockwise rotation projects you forward, and then counterclockwise can actually drag you backwards. Or remember, you could have clockwise direction pushing you forward, cell stops, reorients, and then clockwise rotation can pull you forward as well. So again, just to reiterate, you know, you can have phototaxis and other types of taxis where, you know, you're attracted to different sources. You're attracted in phototaxis. You're attracted to light, so that you can photosynthesize. Aerotaxis, osmotaxis, hydrotaxis. Don't forget, there's many different kinds of taxis. Bacterium are constantly trying to go towards nutrients and towards helpful things and away from harmful things. And that's the whole point of taxis, and that's the whole point of motility. So that really takes us to the end of the chapter. This was a really fun one. I hope you enjoyed it and learned a lot. If you have any questions, throw them in the comment box below, and I will catch you guys next time.
Dr. D, 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 Dr. D.